Good afternoon, everyone. So the title of my talk is Genetic Diversity in Changing Environment. The central goal of the population genetics is to understand what determines genetic diversity. So I'll discuss why genetic diversity is important. Evolution requires diversity to act upon. If there is no diversity, then nothing can evolve. And also it prevents the population from the risk of getting extinct, and it provides a raw material for the population to adapt to the changing environment. One of the very great example is of papered moths, which are of two colors, white one and the dark one. So before the Industrial Revolution, the tree trunk was white in color and the white papered moths were living happily there. But after the revolution, Industrial Revolution, because of the air pollutant, the tree trunk turned to dark, and then these white ones are easily recognizable from the by the predators, and their population started to decrease. Whereas the dark one, they got the advantage and their population started to increase. So this is one of the examples where the diversity helped the population from getting extinct. Now, what affects genetic diversity? Broadly, they can be categorized into the four factors. So there are two factors which increases genetic variation, like the mutation, which is considered as the ultimate source of the genetic diversity. It's a random change in the DNA sequence. Recombination, it's a mixing of the genetic materials. And natural selection, which means that the better fit individual will reproduce more offsprings, that tends to decrease the diversity. And the stochastic force, which is nothing but the sampling noise, and I'll discuss. Usually it's considered more the diversity better for the population. What I mean by stochastic force is, consider a system where the population size is finite. Let's say the food availability is finite. Then the population consists of two types, the dark one and the white one. And the dark one has the growth advantage. Let's say they have a higher growth factor compared to the white one, 1 plus S. Then we expect intuitively that since they are advantageous, so their population will start increasing, and they will start replacing the white one, and eventually they will be propagated into the entire population. But this intuition doesn't hold always, because there can be the case in which the dark one, they reproduces initially, but after that, because of some random chance, any stochastic event, they happens to die from the population, and there is no turning back from the white one to the dark one. So in that case, it can eventually it can extend from the population. So it's a system with the two absorbing state. One is the entire population will be of dark type, and the second one is entire population will be of white type. And the extension happens since even though they are advantageous, but still that extension is happening completely because of the stochasticity. And the same story goes when the growth factor is less, or they are, let's say, less advantageous. In this scenario, we can ask what will be the first passage probability, and if they do so, let's say if the mutant spreads into the entire population, what will be the first passage time? Now, when the environment is constant in the sense that the growth rate is constant with time, so I'll compare two cases. One case when the growth rate is higher compared to the white one, and the second case when it is less than the white one. Then we expect that when they are disadvantageous, they will feel hard to propagate, and the time they will take to hit the boundary will be larger. Whereas this is not the case. Even though if I compare two cases with growth rate high and growth rate low, the growth rate high, they are propagating because they are advantageous, but the lower one with 1 minus S, they can, if they have to hit the x equals 1 boundary, they have to do it very early. Otherwise, they can lose from the population at the later times. This is one of the trajectory. I mean, several trajectories we can look at, but the point is that even though the deleterious one or the less mutant with less growth advantage, they increases to higher frequency, but at the later time they get extinct. So the point is that if deleterious has to reach x equals 1 boundary, they have to do it very early, and the beneficial anyway they have the advantage. So the point which I am making from this slide is that the conditional mean first passage time to hit 1 boundary will be identical in both the case, whether it's a beneficial or it's a deleterious. What will be the implication? If we look at the diversity patterns here, so these are the usual valley-shaped diversity pattern. They will be generated by the beneficial mutant and the deleterious mutant. When they have spread into the entire population, they will be identical in both the case because the time is identical. And this creates a problem because now we will not be able to recognize the mutation, what kind of selection the mutation had, whether it's a positive growth rate or a negative growth rate. So we will not be able to distinguish. So selective sweep is simply means the spread of a mutant into the entire population will not be able to distinguish between the beneficial and the deleterious sweep. So here I will be focusing, so far the story was for the static environment, but usually environment changes. Examples are like global warming, less rainfall, shrinking of the forest, or the seasonal variation in the cases of the plants. And the changing environment, what I mean here is that the growth rate is changing with time and the population size is also changing with time. And I will be comparing this, I will be focusing more on the slowly changing environment, because the rapidly changing environment, we understand it can be easily recognizable, and also the rapidly changing environment, we understand the results will be similar to the static environment as if the population is seeing on an average environment. Whereas the slowly changing environment is more subtle in the sense that it's hard to distinguish one, and also it provides a reasonable good ground for the constant environment assumption to be tested upon. The questions which here we are going to ask is, what will be the effect of changing growth rate on the selective sweeps, which I was mentioning before, and what will be the effect of this changing population size and the changing growth rate on the genetic diversity. The methodology which we are going to use here in the changing environment is the usual backward Fokker-Planck equation, which we use when the system has the absorbing boundaries. So we can write down the probability distribution function, which obeys time in homogeneous backward Fokker-Planck equation. So the difference between the changing environment and the constant environment is now there is an inhomogeneity in time. Like if we calculate the eventual or write down the equation for the eventual first passage probability, the left hand side in this equation 1 becomes 0 in the static environment and the equation simplifies. Whereas in this case, things are a bit complicated. So this has been calculated by our group also, and from Wicker and Hermison. I will be needing the first passage probability because I am interested in calculating the conditional mean first passage time. So since it is conditional, so I'll need this first passage probability. So again, that also follows a time inhomogeneous equation. Now in a slowly changing environment, what we can do is we can use the adiabatic approximation, and we can use the perturbation theory in the sense that we'll write down the mean first passage time as an unperturbed one and the first order correction in it with the n omega, which is a rate at which the environment is changing, being the smallest parameter. Mind you that when I'm using this slowly changing environment, it means that, so there are a couple of time scales present here, the rate at which the environment is changing, the growth rate associated, and the rate at which the stochastic force is acting. So when I'm saying the slowly changing environment, it means the environment changes slowest compared to the other two time scales. Okay, so this was a usual Fokker-Planck methodology to calculate first passage times. So there is one more thing which is known as a semi-deterministic approximation. This is applicable when the growth rate is positive, or 1 plus s for the beneficial one. So idea is very simple. Initially, the mutant frequency is very small, so they will evolve stochastically. Once their frequency increases, then the system will evolve deterministically. And again, once their frequency is very high, it means that the native population has become lowered and their population is very small. Again, we will write down the stochastic equation in the third phase. So, and we will write in the stochastic phase, we will write our usual this Fokker-Planck equation. Here I will write the forward Fokker-Planck equation, and since the frequency is small, we'll take x tends to 0 limit, which turns down to the standard Feller equation here. So the idea is that we'll calculate the solution in the stochastic phase and in the deterministic phase, and then combine the solution by matching the frequencies at the boundaries of these phases. Now, in the the specific model which I'm studying here is the usual, the birth-death process. So again, the population size is constant, and the dark one, they are advantageous or disadvantageous, anything. So the process is a two-step process. In one step, it will give birth to two individuals, which will be proportional to its growth rate, and one of the individual from the rest of the population is chosen to die. So the birth rates and the death rates we can write down proportional to their growth rates, how many number of them are there, and 1 minus I by n with a probability randomly we'll select one to die. So I'll compare two cases in which the mutant appears in the first quadrant and the third quadrant. So it means that the growth rate is the mirror image of each other. Then, as you can see that in the changing environment, the conditional mean first passage time for the beneficial and the deleterious, it started to separate out. As I started that, they were identical in the static environment, whereas now you can see that they have started to differentiate. And in fact, the effect is stronger for some intermediate frequency. And again, when the environment changes very fast, you expect the things will be like the average environment, and where again, it's like a static environment where again there is a symmetry. And the right side figure, again, the genetic variation I have plotted. So you can see the beneficial one over a decade of the environmental frequency. This is for the slowly changing environment. You can see that the beneficial one has hardly changed, where the deleterious has changed significantly. This goes with the fact that the first passage probability for the deleterious, the functional form is very different. It's exponentialiness, whereas for the beneficial, it's lineariness. So the first passage probability is very sensitive for the deleterious one, which is reflected in the conditional mean first passage time, which we are seeing that the deleterious are more affected there. And it can increase or decrease depending upon where the mutant is arriving into the population. Now, if I relax the condition of the constant population size, let's say now we have a changing size also, and the mutations are also recurrent, then it's same as the previous model. The only difference will be, so assumption is that the genome size is very large and the mutation is coming at the independent site. So we can consider this problem as a multiple independent processes are going on. And then if we do so, and again calculate the genetic variation, which we will see that this black dashed is for static environment, whereas the pink one is for the changing environment, where both of them are changing. Now we can do the mathematics to find out that what are these numbers, whether it will decrease or increase. It depends upon where it depends upon that how much phase difference is there between the population size and the growth rate. And yeah, so this is the summary. So the main summary is that the conditional mean first passage time, which were identical in both the case in the static environment, which is known as the Maruyama-Kimura symmetry, that's not preserved in the changing environment. Constant environment assumption is pretty good for the beneficial mutation, whereas for the deleterious mutation, it's not good, and they are affected very strongly. Even for the slowly changing environment, where we have expected that the patterns will be similar or the qualitative nature would be similar, but we find that it's very different. And the future work which we are right now doing, the deleterious mutation, they are very unlikely to spread. So what happens when they get eliminated from the population? So this work is under progress. Thank you.
Thank you. Is there any question from the audience? Okay, please go ahead and ask. Okay. So please turn on the microphone over there.
Hi. Yeah. So I wanted to ask. So you choose in the constant environment, a constant population size condition.
Yeah.
You randomly choose an individual to die off, right?
Yeah.
Is it possible to include? So as I see it, the selection differential is affecting only growth at this point.
Yeah.
Is it possible to include like a selection differential for survival or something and write down another term in the same equation?
I'm not sure whether I understand the question properly, but this selection, so suppose this is a selected mutant,
yeah,
which has, let's say, 1 plus S or 1 minus
right.
S
Yeah.
fitness.
That's affecting its growth.
Yeah.
Right.
Yeah. But so the process is like, so you will be choosing this dark one,
right,
with the probability according to its fitness. So
right,
these are the usual Wright-Fisher model in the population genetics literature. And then the rest of the individual is chosen to die. But yeah. Okay. So the death can be of the white one or the dark one. It can be any of them.
Is there any way to tie it back to something like fitness instead of it being?
Yeah, we are actually. I have not used the word fitness, but
existing. So
yeah, but this dark one is chosen according to its fitness.
Right. Okay.
Yeah.
Any other question from the audience? So if not, I'll ask you one question. So initially you mentioned that there are many of the microscopic forces and one of them is recombination. So I can understand that when you are considering the particular moth example, the recombination may not be as dominant force as a mutation. Mm-hmm. But when you look at viruses, it's a pretty dominant force.
Yeah.
So how will your result change in the presence of recombination? Like what kind of factors do you think?
Yeah, so so again, it's again a competition between the time scales. So the sweep which is doing is reducing the diversity, because that's getting fixed into the population. So it will be hitchhiked the neutral mutant, let's say, associated with it towards the fixation. But recombination can distort it. If your recombination is very high, then we expect that the diversity will again be prevailed into the population. But the sweeps, which I was mentioning, the reduction in the diversity, it's reasonable, or it's good when, or the diversity is significantly reduced when your recombination rate is lower than this conditional mean fixation time. So a competition between these two time scales.
Thank you. Okay, please thank our speaker one more time.